AS Level Biology 9700, Paper 1, Variant 3, October, November 2024. And this is the fourth video on this paper. Now today we will discuss the question number 31 to question number 40. So question number 31 is, what happens in the heart at the start of ventricular diastole? So at the start of the ventricular diastole, it is a phase of the cardiac cycle where the ventricles relax and fill with blood. So diastole, relaxation of the ventricles, and they are ready to fill with blood, which will come from the both atria into the ventricles. So the first option is the semilunar valves open. Semilunar valves will be open at that time when the blood will flow from the ventricles towards the aorta and towards the lungs. So semilunar valve will open at that time. So from the left ventricle, the blood will move towards the aorta, and from the right ventricle, the blood will flow towards the lungs. So at that time, the semilunar valves open. Do not at the time of ventricular diastole. So atrioventricular valves open. Yes, at that time when the ventricles are ready to fill with blood, and we say that ventricular diastole has been started. So first of all, before starting, the atrioventricular valves will open. Statement 3, the pressure in the atria rises above the pressure in the ventricles. Yes, when the atrias have the pressure high than the ventricles, at that time the atrias will push the blood towards the ventricles. At that time, the atrioventricular valves will be open. If the atrioventricular valves are open, at that time the blood will move from the atrias towards the ventricles. So this matches with the B option, but B option is more, you can say, suitable for this answer. The pressure in the left atrium rises more than the pressure in the right atrium. Left atrium rises more than. There is little bit difference in the, but at that time, during the ventricular diastole, we will not consider this answer. Only B is the correct option. We go with the ventricular diastole. The next question is, the electron micrograph shows a longitudinal section through a blood vessel. You can see a blood vessel which has very thin walls, and red blood cells can pass through, and they are very flexible. That is, they can change their shape when they move through this blood vessel. So very thin blood vessel is there, and the option is this is the capillary. So arteriole, why this is not arteriole? It is muscular in nature. It is muscular, and due to which thick walls. But here you can see these are not thick walls. And then capillary is there, vein and venule. Vein and venule, again, they have thin walls. They have thin walls, but their diameter is larger. They have more muscular than capillary. Capillary is only one cell thick, through which only the red blood cells are able to pass through. So the capillary is the answer. Vein and venule are also, they have thick walls, they have thin walls, but their walls are thicker than the capillary, because the capillary is only one cell thick. So B is the correct option. Now question number 33 is there. So question number 33 is, a student drew a sketch to show the formation of a tissue fluid. Which label is correct? So here you can see the blood, the blood enters capillary and pressure increases. When the blood enters into the capillary, then at that time the pressure becomes lesser as comparison to arterioles, because the blood enters from the artery to arterioles and then into the capillaries. So at that time when the blood enters into the capillary, so at that time what happens? The pressure becomes lesser. So A is not the correct option. So at that, second is B, red blood cells and water move into tissues. Red blood cells will not move. Red blood cells will not pass through. So only water will move into the tissues, which will increases the water potential, which will increases the water potential. So this is not the correct option. So when there will be the high water potential in the tissue fluid, so water moves back into the capillary. So C option is there, higher water potential in tissue fluid, so water is moving back into the capillary. So at this venular end, you can say after the at the end of the capillary there is venular end, that is the venules will be there, and venules they will join together to make a vein. So at the here, there the hydrostatic pressure has significantly dropped. The hydrostatic pressure will be dropped because the veins have lesser pressure as comparison to capillaries and arteries. So when the uh, pressure is dropped, the osmotic pressure becomes relatively higher inside the capillary than in the tissue fluid. So this means the water potential in the tissue fluid is higher than the water potential inside the capillary. So due to this water potential gradient, water moves by osmosis from the tissue fluid. So this label actually describes the process of water re-entering the capillary at the at this end, which is called as venule. So C is the correct option. Now D, higher volume of water returns to vein. So the D option, not all tissue fluid, you can say, returns. Some of it remains into the tissues. So D is not the correct option. Only C is the correct option. Now the next question is, which structures are present in trachea and also in all bronchioles? Trachea further divided into bronchus and then into the bronchioles. So here we are going to discuss about that cartilage, smooth muscles, epithelial cells, which are present. So first of all, we will go with the cartilage. So cartilage is present in the trachea. You know, the trachea is C-shaped. C-shaped trachea is there. So cartilage is there, which prevent it from collapsing. And so it is a type of, the cartilage is a type of bone. And then in the bronchioles are very smaller branches of the bronchus. So they are smaller, so they do not contain cartilage. So it is not, the cartilage is not present in them. Second, smooth muscles. So trachea, the posterior wall of the trachea, uh, they have smooth muscles in them. So this allow, you can say, the due to which there is a little change in the structure. So smooth muscles are there in the trachea, and in bronchioles also have a significant amount of smooth muscles in their walls. So smooth muscles allow for the regulation of airflow into the alveoli by constricting or dilating the bronchioles. So smooth muscles are present in both types, trachea and bronchioles, and the third type are the epithelial cells. In trachea, the trachea is lined with ciliated columnar epithelial cells, which have also the goblet cells. So the epithelial cells play a crucial role in trapping foreign particles and moving mucus out of the airway. So epithelial cells are present in trachea, and in bronchioles are also lined with epithelial cells, although the type changes as they get smaller, so but they are present in bronchioles as well. So 2 and 3, uh, smooth muscles and epithelial cells are those which are present in both. So according to this, C is the correct option. A, B, and D are not present over there. Now we are moving towards the next question, and next question is, the photomicrograph shows a section through a part of the gas exchange system. And you know the gas exchange system, uh, where the gas is exchanged as oxygen, which is in higher concentration inside the alveolus. uh, it will move toward into the blood capillary, where the carbon dioxide is in higher concentration, which will move into the alveolus, and through alveolus it will go out of the body. That is the we will exhale over there. So exchange of gases takes place by the process of diffusion over there. Now question is the which type of cells are there, which type of tissues can be seen. So here, is it a cartilage or ciliated epithelium or smooth muscle or squamous epithelial? So this is the alveolus. Alveolus, there is no cartilage over here, because gas has to be exchanged over there. So this is cartilage is a connective tissue. So it is present in the trachea or in bronchi, not in the bronchioles, and in the alveolus it is not present. So cartilage is not over there. So it cannot be the correct option. Then ciliated epithelium. Ciliated epithelium is present in the trachea and in bronchi again. Its function is to trapping dust and pathogens, but it is not present into the alveolus because there should be the exchange of gases has to be taken place over there. Then smooth muscles again. They are smooth muscles are present in the walls of the bronchioles, and bronchioles at the uh, ends of the bronchioles, the alveoli are present. So regulating their diameter and thus airflow. So due to which uh, prominent structures in the image are, you can see they are the air sacs or the alveoli. So smooth muscles are not over there. So only the D option, squamous epithelial, so is characterized by a single layer. So this is the characteristic of the alveoli, that is their internal layer is a flat, consists of flat thin cells which are called as squamous epithelium. So this is the fact, that is the internal lining of these alveoli is lined with flat and thin cells, or which is a single layer of the flat and thin cells. So they are the this type of epithelium forms the walls of the alveoli and the capillaries surrounding them. So B is the correct option. Next question is, what is the function of the goblet cells in the gas exchange system? So goblet cells have a distinct function. So in the gas exchange, specialized glandular cells, which are found in the lining of the respiratory tract. Um so their primary function is related to the mucus protection. So they produce the or release the mucus to increase the surface area. The first, they do not increase the surface area. Surface area is increased by the alveoli. So to move mucus, the mucus is not moved by the alveoli. The mucus is moved in the respiratory tract. So this function is carried out by the cilia of the ciliated epithelial cells. So it is not moved by the goblet cells. So the function of the goblet cell is to produce or release the mucus. So their major function is to synthesize and secrete mucus, which is a sticky viscous fluid. So to trap dust and pathogens, mucus can trap the dust and pathogens, but not the goblet cells. So the goblet cells only synthesize and release the mucus. So C is the correct answer. Next question, which organisms have cell walls that can be affected by penicillin? So penicillin, bacteria. First of all, the bacteria, their cell walls are made up of peptidoglycan, and penicillin, which is an antibiotic, directly affect the walls of the bacteria. So the target of the bacteria is the peptidoglycan walls of the bacteria. So this is correct. So protoctists uh, are the diverse group of eukaryotic organisms. So, but their cell walls are composed of various materials like cellulose, silica, or calcium carbonate. They are not made up of peptidoglycan. So, they are not affected by the penicillin. Only the bacteria, which are the prokaryotes, they are affected by the penicillin, which is an antibiotic. Viruses are not the cells; they don't have cell walls. So only bacteria is the correct option, which is C. Question number 38. So question number 38 is, each year there are 462,000 deaths from malaria in children under 5 years old globally. Insecticide treated nets could prevent 50% of malaria cases and reduce deaths of children under 5 years old by 18%. 18%. So how many children could be saved by using insecticide treated nets? So total number of deaths are 462,000. So out of 462 deaths, there are 18% children. So we have to find out their number. So for this 18%, we have to find out 18% of 462,000 deaths. So we will multiply 18% of 462,000 deaths. So it will be equal to 462 multiplied by 18 divided by 100. So after calculating, let me calculate, it will be equal to 462 then multiplied by 18 and divided by 100. So it will become 83160. So B is the correct option after calculating. So B is the correct option. Next question. Which row is correct? Which row is correct? Involved in phagocytosis. Phagocytosis is a process in which the white blood cells engulf the microorganisms, and beta lymphocytes, they secrete the antibodies. So, beta lymphocyte secretes the antibodies is a correct involved in phagocytes. These lymphocytes are not directly involved phagocytosis, but they can trigger or enhances. So they can release cytokines that attract and activate phagocytes to the site of infection. Additionally, phagocytes can process and present antigens for engulfed pathogens to T cells, initiating an adaptive immune response. So I would like to say that the T lymphocytes directly will not. So it in this regard, it can be the correct option. So T lymphocytes will not secrete antibodies, B lymphocytes not. So only A is the correct option. Already told you they are not directly involved in phagocytosis, but they enhance and activate by secreting other chemicals the process of phagocytosis. So secrete beta lymphocytes, they secrete antibodies. So A is the correct option. Moving towards the last question. 4 steps that occur during the primary immune response to a pathogen are listed. So they have given us the 4 statements. The 4 statements are of the primary immune response to a pathogen. So in this regard, the process of primary response starts when uh, pathogens recognize pathogens recognize and antigen presentation will take place. So then it will happen that the T helper cells interact with macrophages that have digested pathogens. So in this case, the T helper cells will interact with macrophages and then have digested pathogens. So they will represent them. So number 1 is Q. And then second step will be the second step is so when the T helper cell recognizes the presented antigen on the macrophage, it becomes activated. So the activated T helper cells then play a central role in coordinating the immune response by activating other lymphocytes. So at second, there will be the activation of the other lymphocytes, and then when they are activated, then what will happen? This will stimulation. It will stimulizes the beta lymphocytes to divide by mitosis. So the third step is B, and then fourth steps, then plasma cells will start secreting the antibodies. So this is the fourth step. So that is the first step will be the T helper cells interact with macrophages. They have digested pathogens because they will represent them in this way. After representing them, they will become activated. Then they will be activated. Then they will activate the other lymphocytes. Then after activating the other lymphocytes, the beta lymphocytes will start dividing by mitosis. So when they will start by mitosis, so at that time the plasma cells will start secreting the antibodies. So these are the 4 steps in sequence. So these steps can be arranged in the sequence in which they occur. We have arranged them. So which step occurs third in the sequence? So the third is in the sequence is P. So B is the correct answer. So it is in the sequence Q, S, P, and R. So this is the correct sequence. So it was all about from question number 31 to 40. The students, this is the fourth video on this paper. Now I have completed this paper. You can watch other videos as well for this paper. I will discuss more papers for this. You have to like, share, and subscribe my channel. Thanks for watching.